Now, the remaining two terminologies which are going to discuss are mosaicism and uniparental disomy. So, first one, it's the genetic part two, genetics topic part two, and in which we will discuss mosaicism. Mosaicism, or it's like mosaic. Mosaic mean mix. So, in mosaicism, we have one individual having two different, genetically different, two genetically different cell lines. So, the presence of two genetically different cell lines in a same individual, like an individual having more than one chromosome, or more than having somatic additional somates. So in mosaicism, it is actually the presence of a different two different cell lines in the same individual. And the common example of it has two types: somatic mosaicism and germline mosaicism. As germline mosaicism, germline mosaicism. I hope you all are already familiar with the germline mosaicism, as its common example are trisomy 21, Down syndrome, Turner syndrome, Edward syndrome. So in germline mosaicism, the presence of two different cell line two different cell lines in egg or sperm, like in gametes. So the presence of two different cell lines in the cell or gamete, and it's common, and it is a transfer to present in the gametes. It is transfer to the respective generation, transfer to generations. The common examples are Down syndrome, Downs, Turner, Klinefelter, etc. So, if in the question stamp there is no indication of a parental or a relative involvement, then always suspect germline mosaicism. And after germline mosaicism, we have the somatic mosaicism. Somatic mosaicism mean mosaicism in the somatic cells, like the presence of the genetically different cell lines in the somatic cells of the body. And in this example, in this type, we have a common example of McCune Albright. And this somatic mosaicism, it always happen after fertilization. After fertilization, like in the morulation phase, if I remember from the uval diagram, it happens after morulation. So this is after fertilization. And as we discussed about the McCune Albright is a very typical, very common question which we have seen, which I have seen personally in a lot of uval questions and also in NBMEs. So what is McCune Albright? It is McCune Albright. It is actually the mutation or the mosaicism in the somatic cells, which can causing G stimulatory um, G stimulatory protein, which is which is important in cell signaling pathway. So it is the activation of mutation, gain of action. So it is activation of mutation in the GAS or GS, and with this activation of G stimulatory, we have a lot of symptoms, in which the first one is when the GS is overly activated, then it can proliferate the cells, express the cells, increase the products of the cell, so increase or over stimulation of the cell. And in McCune-Albright, we came across from the very much familiar fibroblast like cells. So in this McCune-Albright, we have always unilateral cafe or late spots versus neurofibromatosis, which has bilateral. Cafe spots are also present in neurofibromatosis, but in neurofibromatosis it crosses the midline and it is bilateral, and it has also coast of cornea like smooth edges. It has smooth edges like the coast of California, and in Cafeola unilateral McCune-Albright it has raked edges. Raked mean irregular, like coast of Manet. Then we have Cafeola spots, and we have endocrinopathy, at least one. And this endocrinopathy, we have the precocious puberty, acromegaly, Cushing, and acromegaly, Cushing, and thyrotoxicosis. In McCune-Albright, we have GS activation, especially in the fibroblast-like cells. So if we consider this part, fibroblast, so we have also known the fibroblast is very much important in thyroid gland or thyroid secretions. So in thyrotoxicosis, it is actually the overactivation of the fibroblast which can cause gags, accumulation, and all that features. And this fibroblast link is very much important in thyrotoxicosis. So thyrotoxicosis and Cushing disease and precocious puberty are the most common complication or most common endocrinopathies in McCune-Albright. In Cushing syndrome, we have the cortisol issue, as we know that cortisol decreases the fibroblast activity, which leads to poor wound healing. So that's why Cushing and thyrotoxicosis is very much common in those McCune-Albright. Regarding the acromegaly, I don't have find very much a mechanism about the acromegaly, how this is interlinked, but we can predict that it is a low yield value. So the most common low yield complications, the most common is in 3-1. So there is endocrinopathy, there is Cafeola spots, and there is last one is the fibroblast activity in the bones. So regarding the bones, we have the polyostotic fibrous dysplasia. So this is actually the dysplasia or the abnormal the replacement of the bony bony osteocytes to the fibroblast and collagen, and it is actually due to the activation because of the GAS activation. It is due to the activation of the osteoclast. The osteoclast lead to polyostotic fibrous dysplasia. Bone is replaced by collagen fibroblast, and then this McEwen Albert, we have typically this triad. Then we have two other things, like if this is heterozygous, not heterozygous, actually it is somatic mosaicism is after fertilization, leads to McEwen Albright. But if this fertilization, this mosaicism occur, if somatic mosaicism occur before fertilization, then it will lethal. Maybe before fertilization it may be lethal, maybe it is a severe McEwen Albright, and but if is after fertilization, then this somatic mutation is very much survivable. So this is all about mosaicism and the important parts and important things about this. Next we have after this McEwen Albright, we have this uniparental disomy. So in this uniparental disomy, first we have to discuss about imprinting. What is imprinting? Imprinting means that imprinting is a normal physiological process in which there is a selective gene expression. So gene expression of parent of origin, and so the one copy of the gene and one copy of the allele, one copy of one gene copy is selectively expressed in the other one, and the other copy of gene copy is methylated or silenced. So this is normal physiological process. One gene is expressed and the other one is silenced. So when this imprinting process is aberrant, this imprinting process is abnormal, then we have this imprinting process is dysfunctional, then we have a condition known as dysfunctional imprinting leads to uniparental disomy. If we closely observe the name of this uniparental, uni mean one, parental mean one parental copy of gene. So there is a one parental copy of gene is functioning or okay, or if expressed, and the other one is silenced, deleted, etc. And it is also disomy. So di mean two and somy mean chromosome. So in this uniparental disomy, we have only one parental gene which is functional and having two chromosomes. So how will this possible that from one origin or the n number of chromosome, there is an individual having the 46 chromosome? So definitely this one parental set of chromosome or n, they are duplicated to give to n or euploid, having the two number of chromosome. and having, if the number of chromosomes is okay, then the phenotype is also okay, normal phenotype, right? So in uniparental disomy, there is a functionally there is a disomy, like duplicated chromosome 46 chromosome, which are originated from only one parent, and the other parental set of genes chromosome are methylated or silenced. So this is a uniparental chromosome, uniparental disomy. And then we have hetero disomy. So heterodisomy occur in the meiosis 1, and isodisomy occur in meiosis 2. So disomy has one eye, so this is one. Isodisomy has two eyes, it has meiosis 2. Heterozygous mean heterozygote, and isodisomy mean homozygote. So homozygote is much more complicated, having severe having the severe conditions. Then we have this actually homozygotic and post zygotic after the zygote formation. And there is an important line that if consider isodisomy, always consider isodisomy in an individual which is manifesting a recessive disorder, like a person is having a recessive disorder, having the both small a's or the defective chromosome, then always it give rise to homozygous or isodisomy conditions. Individual manifesting a recessive disorder when only one parent is carrier. So consider isodisomy in an individual manifesting a recessive disorder when only one parent is a carrier, and parent is carrier mean it has this genotype, one parent is. So after this we have two common example, Prader-Willi and Angelman syndrome and Prader-Willi syndrome. So in both occur at the chromosome number 15. Prader-Willi have the problem with the dad chromosome. Prader mean D, so dad is absent, dad chromosome is absent or micro deleted at the chromosome number 15, micro deleted, absent, or there is another condition in which we have no micro deletion but there is always no micro deletion, normal FISH fluorescent in situ hybridization, but it has it's in 25 case 25% of 1/4 of the patient they have the maternal uniparental disomy. So this is very much important MCQ. I have opted this Prader-Willi question, and directly I opted the father and the chromosome option, but it is actually incorrect, and the correct answer is maternal uniparental disomy. So afterward that I keep an eye on such sentences or such questions, that the question might be changed with respect to their uh, high yield criteria. So in 25% a predator will lie, we have been chromosome, a dad is absent, and mom is parental deletion, it has no dad. So paternal or dad is deleted, so the maternally, so the mom chromosome, mom genes are silenced or methylated, right? And in this it has a clinical picture of very much common food seeking behavior, hyperphagia, which leads to obesity. Then we have intellectual disability, obesity, and obesity leads to very much increased level of estrogen, which disturb the gonadal pathway, gonadal axis, or hypogonadism, and then hypotonia. And in angel man we have the M part, so there is a maternal deletion, or mom is absent or micro deleted, and it is almost 5% of the parental of origin, paternal uniparental disomy, and it is also on the chromosome 15. So paternally derived, especially the ubiquitin gene, paternal ubiquitin 3A is silenced, especially this one, and it leads to sale, so sears. And then intellectual disability, which is severe intellectual disability. Then we have ataxia, and L for laughter, like happy puppet. Let's think about how this happened. It is always neuro CNS, it is always CNS. So if we consider the function of ubiquitin proteasome pathway, then these pathways are very much important in degradation of the abnormal proteins, the folded, the misfolded ones. So if that pathway is disturbed, then the misfolded protein, which aggregated in different parts of the cells, especially the neuronal cells, and if these neuronal cells are having the abnormal protein, like we have seen in Alzheimer's, Huntington, and all other the CNS disorder. So if this degradation pathway is disturbed, or is it's happened, there is no gene for the ubiquitin pathway, so there is no ubiquitin degradation pathway. So if this is absent, then it might lead to accumulation of these proteins, which leads to sears, ataxia, intellectual disability, and having the social smile, or like laughter. So this is all about Angelman syndrome. And the next most important clue for this part of the question, the most important clue is fluorescence in cytohybridization. This test detect any deletion, any abnormality in the genes. So if there is abnormality, if there is a microdeletion, then okay, its predator will lie due to the dad or mom microdeletion. But if there is no microdeletion, then the fish is normal. So this clue tells us that whether the question, whether the boards are asking about tell the pathophysiology of the most common like microdeletion, or if the boards are asking about the high yield or the rare one pathophysiology, that is maternal uniparental trisomy. So we have to consider this clue. We have to look for this clue. If this is present, then it is the question is maternal uniparental disomy or paternal uniparental disomy. But if it is not present, then it is definitely imprinting error or microdeletion. So this is all about imprinting and dysfunctional imprinting, uniparental disomy, Angelman and Prader-Willi. So this is all about. So we just wrapped up all the genetic terminologies along with McCune Albright and Angelman disorder.